Hello again. This is the last module in the Forensic DNA Lab, and today we'll be doing the electrophoresis module, which is running the PCR products from the previous lab on an agarose gel. So, a quick overview, the lab will consist of several steps. So first, you will pour a 1% agarose gel, and you'll prepare this gel for running. You will add loading dye to your PCR reaction from the last lab. Then you will load this sample onto the gel and run the gel for about 40 to 60 minutes. And finally, you will image the gel on a UV transilluminator. And hopefully everything goes well and you can rejoice. Um, I, it is my impression that Dylan already poured the gel for you and prepared it. But this walkthrough will show you how the gel is poured. Okay, so the first thing um, you have to do is measure out 0.8 grams of agarose, and the goal is to make 80 milliliters of 1% agarose dissolved in a running buffer. So here Dylan is weighing out the agarose in a beaker. So just fast forward, there we go. Great. So the next thing to do is to measure out 80 milliliters of running buffer, and this is how much you need to pour the gel. And you can use a graduated cylinder for this. And in today's lab, you will be using 1x TAE buffer, but in this particular video, Dylan is using 1x TBE buffer. They're both equally good, so don't worry about that. And at this point, the agarose is not yet able to dissolve in the cold buffer. So this mixture has to be heated up in a microwave. This is what we're going to do here. And the idea is to heat up the mixture up to its boiling point until the agarose is fully melted and dissolved. And Dylan is adding here a glass cover to make sure that it doesn't splatter in the microwave. Here you have to be careful not to overboil it because that will make a mess. And you also don't want to, and at the next step, you want to check after heating it up for a while, you want to check to make sure the agarose is fully dissolved. And to do that, you swirl the hot solution and you check for any little flakes of agarose floating in solution. If you see some, you heat it up for another 10-15 seconds. If you don't, means you now have fully dissolved the agarose. And note that Dylan is wearing gloves to make sure that he doesn't burn himself. This is very hot. Okay. After preparing this solution of agarose, we normally let it cool down for 5-10 minutes until it reaches a temperature of about 60 degrees Celsius. And at this point, we add the DNA stain. And in this particular case, we use CyberSafe. You can also use Gel Red. Historically, people have used ethidium bromide for this purpose. However, ethidium bromide is quite carcinogenic, so it's not recommended to be used in a high school lab. And this stain is at 10,000 concentration. So for 80 ml of gel, you only need 8 microliters of the stain. So you add it to your agarose and you mix well, and now you're ready to pour the gel. So to pour the gel, you first have to prepare the gel box. So here you have the container in which the gel is being poured. You have a comb, which is placed inside the gel container. And then this whole contraption is being mounted in the box, creating a nice seal. So in black here, you see a rubber gasket. And the idea is as you pour the gel will harden in this enclosure and will not leak. So the next step is to now pour this agarose gel, and it will be at a temperature of about 50 to 60 degrees, so you can touch it to your skin to check, or you can use a thermometer to check the temperature. And in this case, the gel seems to have cooled a little too much, so it started to harden inside the beaker. Okay, so at this point, the gel is ready to set, and it can set at room temperature for 1 to 2 hours until it hardens fully. You can also um, prepare it the day before and wrap it in plastic wrap to make sure that it doesn't dry up. You can keep it at room temperature for about a day, or you can put it at 4 degrees in the fridge for a couple of days. Right, so at this point, we have a gel that was prepared the day before. You can take off the wrap and prepare the apparatus for loading your samples. And that entails taking out the gel holder. You have to do this carefully and mounting it in line with sort of the long side of the gel apparatus. So now the next part is to add the running buffer. So again, in this case, it's 1x TBE, but in the lab, you will use 1x TAE. And the idea is to fully cover the gel and then have an extra couple of millimeters of buffer above the gel. And this ensures that the gel will run properly. So the gel should be fully submerged in the buffer, like that. See, and there's probably 3 or 4 millimeters of liquid above the gel surface. Perfect. Right, so after pouring the buffer, the next step is to pull out the comb. So this is the white part. And the idea is that the comb, when taken out, it creates these little wells into which you will be loading your sample. Um, and once this is, if your gel has defective wells, if the wells are collapsed or they're leaking, try not to use those and try to use wells that are intact. So finally, you're ready to prepare your sample. And to do this, you will have 3x loading die. This is a mixture of some salt and some dense polymer that makes it denser than water, so causes your sample to sink to the bottom of the well. So you will take 12 microliters of this loading dye and add it to your PCR reaction. So your PCR reaction will also have a volume of 12 microliters. And you want to mix it really well. And you will do this for every single PCR reaction. And the gel loading dye also contains a blue pigment and a red pigment, which will help you see the sample when you load it onto the gel, but it will also migrate when you run the gel so you can monitor the progress of your electrophoresis. Great. So after you've added loading dye to your samples and you've pipette mixed it, um, an optional but recommended step is to spin down your tubes to make sure that all the liquid goes to the bottom of the tube. And this will make it easier when you're trying to load your samples onto the gel. And by now you should be familiar with this step from the previous lab. So the idea is that in each tube you will have 12 microliters of the PCR reaction and 12 microliters of the dye, so a total volume of 24 microliters. Okay, so before we start loading the samples onto the gel, you want to arrange your sample in the order in which you will load them. So um, your gel has 10 total lanes. We'll use only 8 of them. So in lane 1, you will load DNA ladder, 7 microliters of DNA ladder. So this is a mixture of DNAs of different lengths. So you'll have DNA of 100 base pairs, 200 base pairs, 300 base pairs, and so on. And this will serve as a reference when you are imaging the gel, and you will see this in a couple of slides. In lane 2, you will load 20 microliters of your XX, so a female DNA chromosome 16 reaction, and then 20 microliters of XX DNA chromosome Y reaction. And then you will load the XY sample, chromosome 16, and XY sample, chromosome Y reaction. And finally, you will load the DNA that you extracted from your hair um, with the chromosome 16 primers and chromosome Y primers. And finally, you will load another DNA ladder sample, again, to provide reference during imaging. Okay, so this is the order in which we recommend loading your samples. That way, when you are interpreting the results, it's easy to orient yourself. And your teacher will hand you aliquots of the DNA ladder reference sample, and as well as the PCR reactions that he saved from last week's lab. Okay, so now it's time to load the samples and take your time. Don't rush. It's important that you do this carefully. So you can optionally pipette mix your sample up and down a couple of times and take the 20 microliters and carefully pipette it into the well of the gel. And it helps to place your elbows onto the table to provide extra stability. And here is a close-up of this procedure. You insert the tip of the pipette about halfway through into the well, and you slowly release the contents of the tip. You want to be careful not to poke the tip into the gel, because then it'll be impossible to release the sample. Okay, so once you have loaded your samples onto the gel, you want to connect the gel to the power supply. So you have these special covers that have electrodes, and you want to make sure that the black electrode goes to the black plug, and the red electrode goes to the red plug, both on the gel box and on the power supply. And when the electrodes are connected, you can start running the gel at 100 volts. And generally, we would recommend running it for about 60 minutes. But if you're in a rush and you need to finish the lab, running the gel for as little as 30 to 40 minutes should be enough to see the products. Okay, so as you're starting to run the gel, it's helpful to make sure that the gel is running in the correct direction. And so the correct direction means that this purple dye will be migrating away from the well down the gel. So this is migrating in the correct direction. If your dye is migrating in the wrong direction, it means that you must have mixed up your black leads, your black electrode and your red electrode. So you need to double check how you plugged everything in. And finally, after 40 to 60 minutes, you're ready to visualize your gel. So stop the power supply, take off the cover gently. And now that the gel is fully disconnected from the power supply, using gloved hands, take out the gel together with the container with this plexiglass enclosure that holds the gel. And you can dab the extra buffer on a piece of paper towel like this to make sure that it's not dripping. And then carefully slide the gel onto the surface of the transilluminator. So this is a UV light source, which will be used to illuminate the gel and to visualize the DNA that's in the gel. And the 1% gel should be quite sturdy, but you still have to be careful. And next, you want to remove any sort of air bubbles that are trapped between the gel and the glass. Center the gel onto the transilluminator. And place this there. Just center it in the middle. And then place this wooden um, gel imaging box on top of it. So it's simply an enclosure that blocks the UV light from illuminating you and has a special filter which allows you to view the product, to view the gel. So um, you can use a cell phone to do this. In fact, you have to use your cell phone because we don't have any other cameras for this. And the cell phones are actually quite good at capturing the image. And I would recommend taking a couple of pictures because sometimes they turn out of focus. And now we're ready to visualize and interpret the results. So a couple of weeks ago, I ran a test of this experiment before I shipped the reagents to your high school, and I want to walk you through the interpretation of the results. So this is a picture of the entire gel here, which I converted to black and white, and then I zoomed in onto the part of the gel that shows the PCR products. So I want to point out that here you see on the sides, you see the ladders, and my ladder is a little bit faint, but in your case, if you load 7 microliters, the ladder should be much more clear and visible. So here we have the 100 base pair reference piece of DNA, 200, 300, 400, and 500 base pairs. And just to remind you, the chromosome 16 primers should give us a product of about 271 base pairs, and the chromosome Y primers should give us about 313 base pairs. Okay, so um, here I had in the first two lanes, I had chromosome 16 and chromosome Y uh, PCRs using Dylan's hair DNA. So DNA extracted from hair using the Kelex protocols, just like you did. Then I had my DNA extracted from a mouthwash. Again, chromosome 16 and chromosome Y primers. Then I had Dylan's DNA extracted from a mouthwash and Juliet Hammack's DNA, Miss Juliet Hammack's DNA extracted from a mouthwash. So all this DNA extracted from mouthwashes, there tends to be a lot more DNA in it. So this serves as a positive control, and this is what you had as well in your lab. And it is immediately clear that we have very strong, clear PCR products here. For chromosome Y, we have a product that's slightly shorter than 300 base pairs. Sorry, for chromosome 16, it's slightly shorter than 300, and for chromosome Y, it's slightly longer than 300. So this is my DNA, so I have both chromosome 16 and chromosome Y. The same is true for Dylan's DNA. And I want to point out that there's a couple of extra bands here that are quite faint. There's one here, there's one here, there's one here. These are non-specific bands, which result from our primers misannealing, binding erroneously to other pieces of the DNA that are not on chromosome 16 or chromosome Y, and yet we still get faint products. When we look at Miss Juliet's DNA samples, we have a very strong and clear band for chromosome 16 and a very faint band in the chromosome Y reaction. Actually, there's 3 faint bands, and these are all non-specific products, and they're much dimmer than the principal product. Oh, sorry. Um, okay, so the control DNA worked really well. When we looked at the Kelex extracted DNA, we see a clear product for chromosome 16. And however, we do not see a product for chromosome Y. So we know that Dylan has a chromosome Y because um, when we have enough template DNA, we clearly see a product. But there was just not enough DNA here to give us a product, and potentially we could have improved it by maybe extracting the DNA for longer or maybe using more hair follicles. But at least we have one product that worked, and these control products also worked really well. Okay, so this is the end of the Forensic DNA Laboratory series, and we hope that at least some of your PCR reactions worked, and in the best case scenario, that your Kelex extracted DNA PCR worked. But even if they did, sorry, even if they didn't, um, hopefully you had fun throughout. And I want to say big thanks to your teacher, Dylan, who spent his summer working in our lab and developing this protocol and troubleshooting it and optimizing it and trying different primers until we got it to a point at which it worked fairly reliably. And I also want to thank Miss Juliet Hammack, who is another science teacher from Oak Grove. She worked on a different lab, on a protein purification lab, but she graciously volunteered her genomic DNA as a control for your experiment. Thank you very much.